Welcome back. We're going to talk about some more macroevolution. Hopefully you're with me for part one. In part one we looked at phylogenies, which were ways to represent the results of evolution, the results of speciation. In this part we're going to talk about mechanisms of speciation. How the heck do we get new species? We're going to talk in particular about a process called allopatric speciation. But before we can even do that, let's talk about what it means to be a species. So I want you to think about what that word means to a biologist. To say two individuals are part of the same species, what does that mean? Okay, be thinking about that. Let's also look at allopatric. The term allopatric here, what does that mean? We can translate it into English, make it a little simpler. Allo refers to different. Translates into English from Latin as different. Patric means father. Think about the Spanish word padre. You probably know that means father. So allopatric literally translates as different father, but in this case we're using it to loosely translate to mean different fatherland or different homeland. So allopatric speciation, you're going to see as we go through, always refers to population diverging into different locations. So we have subgroups of an original parent population that are physically separated. They are geographically separated, meaning they now have different homelands. Okay, so that's what allopatric means, different homelands, not in the same place. Sympatric, S-Y-M, would be in the same homeland, and that's a different presentation altogether. Okay, so we're looking at macroevolution, which means speciation, and we're going to look at the specific events of allopatric speciation. But first again, what the heck is a species? Okay, look at those slides. You can pause at any point and think about what you see on the slide, take notes. So here is a biological definition of species that's most commonly used today. A group of organisms that have the potential to produce viable, fertile offspring in nature. So by viable, I mean the offspring survive, and they can in turn reproduce and make more offspring in the future. Macroevolution is what we're talking about today, and macroevolution generally refers to producing new species. Enough changes that there are new species produced from the parent species. It could also refer to simply producing complex structures like an eye, but for our purposes we're going to use macroevolution in the most common sense, which is developing new species. So macroevolution and speciation will be interchangeable for us. It says here, in contrast, as microevolution, which refers to smaller evolutionary changes, like changes in allele frequency. But I want to point out that macroevolution is simply the summation of, the accumulation of many microevolutionary changes. So we're not talking about entirely new mechanisms. The mechanisms are the same. We're still talking about natural selection, genetic drift, all five of those that we looked at in earlier episodes on microevolution. So macroevolution has occurred when we have accumulated enough microevolutionary changes that we could designate something as a different species than its ancestors. Okay, so hopefully that's clear. Not different mechanisms, just maybe different time scales, and the results determine whether we call it micro or macro evolution. There are always exceptions to any of our rules in biology, so the term species is hard to use when we talk about organisms like bacteria that don't have sex. Asexual reproduction makes it a little trickier to define species in the typical biological sense. It's also hard to use the definition of reproducing and producing fertile offspring when we look at fossils. So sometimes we have to rely on other criteria. So an interesting aspect of studying species in evolutionary terms is looking at species that are connected geographically and connected in terms of reproduction, but can still result in new species along the way. And I may not be very clear, but let's look at some examples here, and hopefully it'll become clear. We're talking about a phenomenon called ring species. So ring species are a group of individual organisms that are connected, or populations, I should say, populations of organisms that are connected side by side by side geographically. Let's look at this picture. So look at these salamanders in California. So the salamander at the top is connected geographically to the one on each side of him, so their ranges overlap. And then this one's range overlaps with the next one, which overlaps with the next one. On this side, his overlaps with his overlaps with his. So we call each of these could be considered subspecies. They also use the term ring species. And so what do we mean here? It means that Picta, the first one, could share genes with Platensis, the next one on the right. It could also share genes with Oregonensis. And so the different subspecies can swap genes with the guy they're next to. So this one can swap genes, but normally they can't at other extremes. So Picta at the first could probably not breed with the one at the end. But it's still, in a sense, sharing genes by way of each of the intermediate, the little bridges along the way. So another way to think of ring species, it's a way of looking at speciation captured in time. We can see stages of speciation through geographic isolation that might occur through geographic isolation by looking at the changes as we go along the steps of the ring. So here's another depiction of that. We have gene flow, meaning genes are carried from one subspecies to the next, but they can't be carried from one end to the other. If we took an individual from one end, it probably wouldn't mate with an individual at the other end. And in this case, the salamanders at the end of the range don't mate with one another. So by definition, they would not be the same species. So here's another way to look at that ring species. This is a really good diagram, maybe a little clearer than the previous one. So you should pause it and take some notes on what you see here. So let's look at some other ways to notice differences between species and how we would determine whether organisms are in the same or different species. So these are sort of like exceptions to our rule about breeding. If they don't commonly breed in nature, then we would consider individuals separate species. So the Canis genus is a great example. Canis is the genus that your pet dog would be in. Its full name is Canis familiaris, the familiar dog. But we have other members of Canis. Canis lupus is the wolf, and you see other varieties of the Canis genus here in this picture. They don't breed with one another, even when they're in the same place in nature. But if you put them in the same location as a dog, they will often interbreed. But in nature, it doesn't usually happen, so we do consider these to be separate species. Same idea with lions and tigers. They can produce hybrids like the pictures on the right, but it doesn't happen in nature, only in captivity, so these are separate species. If you're with me for our other discussions of evolution, you already know these terms. I'm just going to ask you to pause it here and make notes if you don't remember the difference between anagenesis and cladogenesis. And now we get to the crux of this presentation. We want to specifically talk about allopatric speciation. So on the left side of the picture here, figure A, we have an illustration of how allopatric might occur. We have a parent population that shares the same range, so these pine trees could swap genes all across. They could have pollen spread from one tree to the other and cross the entire range. But over time, maybe something occurs to separate them into two groups. In this case, we have a canyon that's cut, and now we have two completely separate groups of trees. They are geographically separated, and once they're geographically separated, they're not going to be sharing genes with one another, so they would be treated differently by natural selection. They could accumulate different mutations. So the mechanisms we talked about for changes, natural selection, genetic drift, they would be different in the two subpopulations. So that sets up the potential for allopatric speciation. It doesn't guarantee that would occur, but at least it makes it possible. So you can think of it this way. For speciation to occur, you have to have a way to separate the gene pools of two populations. You can separate them physically, that's allopatric speciation. we'll look in a separate presentation about how you could separate them genetically, even if they live side by side, still prevent them from mating and result in speciation. That's called sympatric. But again, that's a separate presentation altogether. So again, allopatric speciation, the first step, and I tell my students there has to be a crucial event occurring to make it possible for speciation to even begin. And that event by itself doesn't guarantee speciation, but it sets up the possibility. And the first event has to be a separation of the gene pool. If it's a physical separation by geography, that's what we have here in this case, allopatric, so that's the first step. Geographic isolation occurs, which just means we took a population and split it into two groups. So imagine a group of squirrels living at location I'll call location A, and they become a large population, and some members start to migrate around this mountain, and they live on either sides of the mountain. So we have some members of the squirrel population living where I'll say X, and some living where I'll say Y. And while they're in those two separate locations, again, natural selection is probably different on each side of the mountain. So traits that are favored on the Y side are not the same as the traits that would be favored on the X side. We also have a little bit of genetic drift, which split this population into two subgroups, and now we have a little founder effect going on. They probably aren't representative of the original population from the very moment that they've split up. And so we have sort of genetically pushed them away from each other in a sense, and the environment has. But the big key here is that natural selection will be different on each side of the mountain. So they'll start to accumulate different varieties over time via natural selection. If they accumulate so much differences that by the time they eventually migrate back together, thousands of years later maybe, and the two populations meet once again, if they can no longer breed with one another, then we would say that speciation had occurred. If they can still breed with one another, then we say speciation had not occurred. Okay, so on the right we see speciation, on the left speciation didn't occur. Okay, now think about the ring species. Ring species again are a way of sort of taking a snapshot of what you see here in progress as the populations are migrating into different locations around some physical barrier and forming that ring that we talked about. Okay, do you want me to pause this one? I'm not reading it to you. So why does speciation occur after geographic isolation? I've sort of described it already, so I'll let you pause this and jot these down. Got it? Hopefully you did. Now I want to look at a specific case of allopatric speciation that is the most well-known way of describing or looking at speciation. It's probably the most common type of speciation that occurs allopatrically, and it's called adaptive radiation. So I want to first describe what this term adaptive radiation means. It's in two words. Think about what those two words mean to you, adaptive and what does radiation mean. So adaptive, probably pretty obvious, becoming adapted to the environment. But radiation here has a lot of different connotations. So when you think of the word radiation, you might be thinking of X-rays or the rays of the sun or gamma radiation. What we're talking about here is simply to spread out. Think about the ways the rays of the sun spread out across the earth. They radiate. So here the term radiation means to radiate, to spread out. Adaptive radiation, quick definition, is when a population spreads into new environments. That's the radiating part of the name, spreading into new environments. And because the environments are now new, natural selection will cause it to become adapted differently in the new environment than it was in the old environment. Okay, so spreading into new environments and becoming adapted. We'll look at how that happens as we go along, but I want you to be sure you understand the terminology. So it's actually sort of named backwards. Radiate has to happen first, and then they become adapted. Okay, so spread into new environments, then become adapted to the new possibilities in that environment. One of my favorite examples of this, it's not going to be in the slide, so I'll just give you a quick example. Think about Hawaii. What do you picture when you think of Hawaii? Maybe you live in Hawaii. Hawaii is a beautiful ecosystem, one of the most lush environments you'll find on the planet, really rich places for life to exist. And think about the kinds of flowers and fruits in Hawaii. Is there a lot of diversity or not much diversity? Of course, there is lots of diversity. Lots of flowers, lots of different fruits in Hawaii. Now, at some point in the distant past, fruit flies were introduced to Hawaii. A single species of fruit fly was brought to Hawaii. Now think about fruit flies. They love to live on fruits. Think about all the different kinds of fruits existing in Hawaii. That provided many, many new environments that the fruit flies had never existed in before. So the offspring of the arriving fruit flies, the founding population of fruit flies, their descendants could spread into all these different environments. Natural selection was different for each type of fruit. So the traits that were favored in the populations on each of the different kinds of fruits on the island were slightly different. As a result, today there are more species of fruit flies alive in Hawaii than the rest of the world combined. Many, many, many times more species of fruit flies in Hawaii and in the rest of the world. And it's specifically because of adaptive radiation. So the population radiated into all these different environments and then became adapted to those different environments. Adaptive radiation. Now let's look at another even more classic example of adaptive radiation, and that involves the finches in the Galapagos Islands. Here's a family tree of the finches in the Galapagos. The finches have ancestors on the South American mainland. The ancestral population migrated out to the islands some 500 to 600 miles away and then spread into many different environments across the many islands of the Galapagos. And within each island there are different niches available, different environments within an island, and obviously from one island to the next the environments are very different. So as the finches radiated into all these different environments, different features were selected by natural selection, different adaptations arose, and as a result we now have 14 different species of finches, all descended from the South American variety. A great example of adaptive radiation. The next several slides are going to explain that to you step by step. They do a great job of doing that, so I'm not going to read it to you, but you should pause as we look at each slide, read it, make sure you're clear on it, take a few notes. So here we go. Now it does say one would expect each island to have only one species. You might expect that. You might expect an island to have multiple environments within a single island, but the simplest answer would be to have each island have its own unique species. But in fact, some islands have multiple species because of the multiple environments within an island. That's what the island looks like. Big map. Now here's the process of adaptive radiation, step by step. Again, you should be pausing each of these. And that wraps up that little piece on adaptive radiation. I want to take a moment and explain one term here for you, character displacement. Character displacement refers to two very similar species trying to live in the same niche, meaning they're in the same physical location, trying to do exactly the same role in nature, eat the same food at the same time, reproduce at the same place. Those would be common niches, and when that occurs, when two species are competing for exactly the same niche, there's only a couple options that can occur. They're not going to coexist in that niche because one of them is going to inevitably be slightly better at occupying that niche, and therefore natural selection will result in its success over time, and the elimination of the species is not quite so good. So that's one possibility of competing for the same niche, is extinction of one species. But another option is for the second species to occupy a slightly different niche. Still in the same maybe physical location, but maybe feeding on slightly different food sources, or maybe living in a slightly different area within that habitat. Think of birds living in a tree If they're competing for food at the bottom of the tree in the forest undercover, it might be possible for one of those to migrate up and feed on insects within the branches of the tree. That would eliminate that competition. Natural selection would then be a little different, and the features that are favored in the one that's now in the middle of the tree would be different from the features favored at the bottom of the tree. So their characters, meaning their traits, get spread apart. They get displaced and become slightly different. Okay, so that's the second option of competition for the same exact niche. The bottom line is that ecologists, evolutionary biologists, believe that it is not possible for two different species to occupy exactly the same niche. We call that the competitive exclusion principle. Okay, you should write that down. The competitive exclusion principle says that no two species can occupy exactly the same niche. A little bit more on the finches. Now, you certainly don't need to know all 14 species for the AP Bio test. It's just a great example of adaptive radiation. It would be good to be able to describe the process of adaptive radiation using finches as an example. And just in case you're curious, here are the 14 species of finch. One of them is not in the Galapagos Islands. It's in a nearby island called the Cocos Island. And on that single island, Cocos Island, we have a variety of finch that has evolved there. It's from the same ancestral population called the Cocos finch. Now here's a great example of natural selection that's been documented within our lifetimes. Doing studies in the Galapagos Islands for over 20 years, a husband and wife team from Princeton University named Peter and Rosemary Grant, they're to this day still studying finch populations in Galapagos. They went to a particular island in the Galapagos where there was a variety of finch called the medium-beaked ground finch. This finch eats seeds that it finds on the ground, and it uses its beak to crack open the seed and get at the food that's inside the seed. They did an interesting study and found that over time, depending on whether it was a particularly wet year or a dry year, the depth of the beaks changed. The average depth of the beak. So no individual bird was changing. I want to make sure you're clear on that. Birds didn't grow a different size beak because of the environment. But imagine a whole large population of birds with a range of beak sizes from small to large within this group of medium beak ground finches. Depending on the environment, which variation of beak was more successful was different. So in a normal year, when there's plenty of moisture around, the plants are producing lots and lots of seeds. Some of the seeds are very large and hard, some of the seeds are very small and soft, and some are in between. So as long as there's lots of water, there's a wide variety of food, and all the finches get to eat. Doesn't matter if they have a big, strong beak or a smaller beak that's not so strong, they can still find food that is that they're able to acquire. But in dry years, when there's a drought, during droughts, the plants are not producing as many seeds. Because they're not producing as many seeds, there are fewer of all kinds of seeds available. Because of that, most of the birds don't have beaks that are strong enough to crack open large seeds. The large seeds are harder and require a larger beak to crack them open. And normally even the large-beaked birds will eat smaller, softer seeds because they're simply easier to eat. But when the smaller, soft seeds are not available, they can still survive by eating the large seeds, whereas the birds with smaller beaks can't make it. They simply die out. They're not very successful because there's not any food left for them. Once all the birds eat all the small seeds, then they're forced to all try and eat the big seeds, and only the birds with the large beaks, with a greater beak depth, have a strong enough beak to crack open those hard seeds. Because of that, in the next generation, there are more genes passed on for larger beaks. So in dry years, the beak depth increases in the Galapagos Islands. So if two populations have diverged and we think they're now different species, remember our definition would say that they couldn't mate. So what is it that guarantees they can't mate? What is it that keeps species separate from one another so that their genes don't just mix back together and revert to some other form. So there are various mechanisms in nature that serve to maintain species as separate species. You can think of it as the how of speciation. It's what guarantees they really can't share their genes anymore. So we have a group of these in two categories. The first is going to be called pre-zygotic. We'll look at these individually. But if you just break that word down, you'll figure out what it means. Pre certainly means before. Zygote. You know what a zygote is? Zygote is when a sperm and an egg unite. So here we're representing sperm and egg uniting. If the sperm and egg unite, you form a fertilized egg called a zygote. Most of the barriers to speciation prevent a zygote from ever forming. The sperm and the egg don't ever get to unite. So that guarantees species are separate if their sperm cells and egg cells don't have a chance to unite. And the barriers that prevent that isolate them reproductively, are called pre-zygotic barriers. But in some cases, two different species are able to make it past all of those barriers, and they do put sperm cells and egg cells together. And if the sperm cells and egg cells join and form a zygote, that still doesn't necessarily mean the individuals came from the same species, because there might be something else that prevents them from producing viable fertile offspring. And those barriers are called post-zygotic. These are barriers that isolate individuals after they've even made a zygote. Okay, so terms that are really important to know, and you should be able to explain each of these mechanisms as being pre-zygotic, before the zygote stops the zygote, or post-zygotic, even though they made a zygote, they're still different species. Okay, so now these slides are going to do a great job of explaining this to you. Lots of text here. I'm not going to read it to you. I want you to just pause each one of these, read it, jot down notes as you need, and here we go. Okay, so bottom line on ecogeographic, they live in different places. Okay, pretty obvious. If two species are in different locations around the world, they're never going to mate. Did you get this one? Habitat isolation at first sounds like ecogeographic. You might think living in different parts of the planet would be different habitats. That's not really what we mean. Here we mean they're in the same geographic location, but different habitats within that location. A good example here would be these frogs. They might live in the same forest, but one reproduces in water of a stream, whereas the other reproduces in shallow pools of rain. Because they're laying their eggs in different locations, they would never ever breed with one another, even though they live in the same physical area. Okay, so that's habitat. Seasonal, this one's pretty straightforward. Pause it and jot down the information. I'm going to give you one more term for it, though. You might occasionally see this called, in fact your textbook might call this temporal isolation. Temporal refers to time. So temporal or seasonal isolation, mating at different times of the day or different times of the year. Behavioral isolation, pretty straightforward. You might be familiar with this. Think about courtship behavior. If the male doesn't do the right behavior, the female's not going to mate with him. Has to act just right. Mechanical isolation, let's just say the parts don't fit. You know what parts I mean. Gametic isolation, this is the last of the pre-zygotic barriers. So imagine that two species have overcome all of the others. They get together in the same place at the same time, mate, do the right behavior, mate, put sperm and egg together, and they still don't mate. And they still don't produce an offspring. So there are factors that keep sperm and egg from different species from recognizing one another, and therefore the sperm cell doesn't enter the egg cell. That's gametic isolation. Don't you love that picture? So here we're looking at post-zygotic mechanisms. They prevent species from being considered the same species, because even though they make a zygote, something else still happens. So I'll let you read each of these. This is the classic example, the one you're probably most familiar with, the one that I've seen on the AP Biology test more than any other. For this one, know both terms, hybrid elimination or hybrid breakdown. So in this one, think about, and let's use some genetics terms. Two parents mate and produce an F1 generation, and the males and females of the F1 are able to mate and produce an F2, a second generation, but then they can no longer mate. They've accumulated so many harmful traits by trying to combine two different species that they are no longer able to mate. And so the end of the line for this hybrid, this crossing of two different species, is in the second generation. So that still means there's no gene pool carried on over time. So as a result, we consider these to not be the same species. So here's a summary of the mechanisms that stop zygotes from being formed. It's a really good diagram. Take a minute, copy it down. And this one summarizes post-zygotic barriers. So these species overcame all of those pre-zygotic barriers in order to put sperm and egg together, and we still don't call them the same species. That's it. I hope that was helpful. We'll see you next time.